There we go. Now we went over the basics of chapter 1. We're going to fly through chapter 2, so make sure you don't fall asleep. All right. So selected slides from this huge PowerPoint that I had before in chapter 2. What you need to know: energy is the capacity to do work, put things into motion, or store some energy as potential energy so you can do work later. Okay? Energy does not take up space. Energy cannot be created or destroyed. It can be transformed. Okay? Then energy is a capacity to do work. Energy forms can be interchanged between kinetic energy, that is energy in motion, or potential energy, that is energy that can be used later, so stored energy. Now, energy can go from one form to another. When it goes from potential to kinetic, then you get some movement out of it, because kinetic means motion. Okay. Now, when stored energy gets released or gets used up, the transformation of this energy is not 100% efficient. Some of it is always lost as heat energy. Okay? All right. So, types of energy that you can have or you can store. You have chemical energy, and that is stored within bonds of chemical substances. You can have electrical energy, and well, we all know what electrical energy is, I hope, right? Mechanical energy is something that has to do with kinetic energy. Okay, so mechanical energy. And then you have this electromagnetic or radiant or even radioactive type of energy. Okay, electromagnetic, it means something to do with electricity and magnetism, including light energy. Right? Remember, light is a form of energy. Okay. Electromagnetic energy travels in waves. Okay. What are the forms of electromagnetic energy? So you have heat, you have visible light, you have light that is not visible, you have radio waves, X-rays, gamma rays, microwaves. Right? This is all forms of electromagnetic energy. All right. As I mentioned, when you convert one form to another, you don't have 100% efficiency of conversion. You're always going to lose some energy in this conversion process. You're going to lose it as heat. All right, which of the following is an example of the conversion of potential energy into kinetic energy? B. That is, you break up bonds that are in ATP to get some movement out of it. So the bonds in the ATP molecule, those are stored energy, and the movement of muscle contraction, that is your kinetic energy. So flying through the periodic table, and I'm not going to ask you much about the properties of the periodic table, but there are some things that you should know, because it should be, very, very intuitive and second nature, okay? So let's look at these guys here. This column here, those are called noble gases. They do not react, and they don't like to interact with anybody. They have the nose up in the air. They are noble, right? That means the outermost shell of these atoms is totally filled up, and they don't need to pick up electrons or donate electrons. They don't react. All right. And then you look on the other side here, you have certain atoms that are not filled up, right? And they can be very much reactive. Okay, so in this case, they have like one electron in the outermost shell. We're not going to go into a lot of details about that. Now, some of those elements here, some of the elements that are contoured here in black, those are the elements that will make most of the molecules in your body. Okay, so what are they? Nitrogen, oxygen, phosphorus, sulfur, and of course carbon. Why isn't carbon highlighted here? There we go. Okay. Carbon is a very, very important one. All of organic chemistry is based on carbon. A lot of fun, right? What are the atoms that are making most of living things? Those that are here, I mentioned those. And then you have also calcium, potassium, sodium, and hydrogen. Okay, so here are the elements or atoms that are really, really important for life. And of course, carbon is at the center of life forms. Okay, so looking at things that get dissolved, you have your solvent. In the case of living things, your solvent is always water, right? There is no other form on earth that has other solvent, so that's water, okay? And then you're gonna have stuff that is either totally dissolved in the water or is suspended in the water, okay? So if you get stuff that is totally dissolved in water, you call that a solution, right? So your solvent is the water, and your solute is whatever you put in the water to get dissolved. So let's say, for example, table salt, NaCl, that will be your solute, and water is your solvent. Okay. And then you have other forms of mixing up things that are called like colloid. They're not totally dissolved, but they're not in suspension, and they kind of change the properties of this solvent. Example given here is like when you make jello, okay, gelatin, right? So you have some particles that are floating, and they interact enough with the water to change the properties of that solvent. All right. And then you have another case, the suspension, and this is really cool because that has a lot to do with physiology. Because when you look, for example, at blood, you know that blood is mainly plasma, which is water-based, but you have a lot of stuff that is floating in the blood, like your blood cells, white blood cells and red blood cells, and all the platelets and everything else that goes with it. Okay, so if you spin and centrifuge this blood, you are going to get separation of whatever was suspended in there. You're going to get separation, that the heavy stuff is going to go to the bottom. Okay, so that tells you that if you can separate, the thing is a suspension. Now, if you try to separate something that is in solution, what's going to happen? Like exactly, like nothing, right? Nothing's going to happen because in solution means totally dissolved. It's not going to be heavier and be able to separate. Okay. All right. So here is just what I have talked about. Okay. So when you talk about mixtures, right, you mix things in the water. So chemical terms for when you dissolve things in water, you usually use these terms that are involving molarity. Okay. So molarity, or big M, is the number of moles of solute per liter of solvent or water. Okay. So this is why I have a little mole over here in a liter of what's going to be water. Okay. What is a mole? Is a number, just like a dozen is a number. How many things in a dozen? 12. How many things in a mole?
6.02.
There we go. That's Avogadro's number. Avogadro's number, no. Avogadro's number is 6.02 times 10 to the 23 molecules. I mean, you could call it a dozen, but it's a lot more than a dozen, right? It's just a number of things that are standard that dissolve in water. Okay. All right, so we talk about colloids. That's a thing that makes jello, and they are also known as emulsion. Not emotion, emulsion. Okay. Okay. Now, I really want to get, I already talked about suspensions. I really want to get a little bit about the water. Okay. Everybody heard of water properties before? Yeah. We know water has a dipole because oxygen likes to grab electrons more than hydrogen does. So oxygen is more electronegative. It grabs the electrons, right? Now, in order for water to be in liquid form, it has to form hydrogen bonds, right? And that's very important for maintenance of life, okay? So how strong is a hydrogen bond? Well, it's not very strong. if you compare with covalent bonds that are bonds between molecules, so hydrogen bonds are not strong at all. They're stronger than the other bonds that are called van der Waals interactions. Those are gases interactions that are even weaker. But hydrogen bonds are strong enough to sustain life. Water is a polar molecule. Oxygen is electronegative and hydrogen is electropositive. What does that mean? Which one pulls electrons? If you're pulling electrons, you're becoming more negative. So it's not D. It is C. All right. Electrons, oxygen pulls electrons away from hydrogen. All right. Okay. All right. So now flying through chemical reactions. We hopefully have seen before some chemical equations. Here is a representation of a chemical equation that can go both forward or backwards, right? And we see here we have two molecules of water, of course represented in the classical H2O state, right? So we see here that they can interact and produce a charged molecule and that is positive and a charged molecule that is negative. So this guy here is called what? Hydronium ion, erroneously called a proton. So that's a hydronium ion. And this other guy is what? Hydroxide, right? So you have some charge separation here when you look at the interaction of water molecules and this chemical equation. Now, what you have to know, of course, you have to identify a chemical equation, especially the ones that are in relation to bodily functions, because normally those are equations that can be reversed, right? And what do you have in those equations? You're going to have things that go on your right side, and those are your reactants. And you're going to have things that go on the left side, and those are your products. Okay? And when you look at reactions that are happening in the body, usually the thing that allows this reaction to take place is some kind of protein, and that is a catalyst. Okay. Types of reactions. Here you have one that is showing a simple decomposition reaction. So you have a compound that breaks into two parts, into elements, right, on two different parts. So if you're breaking things down, that's a decomposition reaction. And this arrow just going forward is telling you that this is an irreversible reaction, which is just to exemplify that this is decomposition. As an example, we have here a molecule of glycogen, which is this huge molecule that is a polymer, right? Polymer of glucose molecules. And what we use this for is that we use it for storage of energy. So you have a couple of places that you can make glycogen, like for example in your liver and also in your muscle cells. And here, let's say when you need the glucose, you're going to break down this glycogen molecule into individual glucose molecules. So this is an exemplary decomposition reaction. And this could also be called a catabolism reaction. So catabolism breaks things down, anabolism builds things up, and you put everything together and you call that metabolism. Okay. Now, we have other types of reactions that cause exchange of parts of molecules, and this is also called displacement reactions. So you see here that you start up with some compounds, you end up with different ones, and molecules were exchanged. Now, when you look at an example of a reaction, you see here the activation of a glucose molecule. Now, when we talk about activation reactions in biochemistry, we usually mean adding a phosphate group to a molecule. Okay? Now, where is the phosphate group going to come from? Well, you have to look at where is it in the cells that we store energy. So we know the currency of cells for energy storage is ATP, or adenosine triphosphate. Right, so here is a molecule of ATP, and we see that this phosphate group that is further away from your ribose, that phosphate group is the most energetic one. And when you have a displacement reaction, you will see that this phosphate group was transferred from the ATP to this glucose molecule. So now the ATP lost the phosphate, became ADP with diphosphate, and your glucose gained the phosphate, so it became glucose phosphate. So this is an example of displacement reaction. Now, just a little bit more on this chemical equilibrium type of reaction, right? We know that most reactions in the body can go either forward or backwards. Now, what is going to determine whether a reaction is going to go this way or that way is really going to be several things. Like the first thing is really like, what is it that the cell needs? Does the cell need this kind of product, or does the cell need that kind of stuff available? Then you're going to say, okay, that's one of the first requirements. Second requirement would be how much of the stuff you have in the first place. So if you have a lot of it and a lot of B, then it's easier for these things to hit each other and become AB. So concentration of products is a very important factor to determine whether you have a reaction that is going to go forward or backwards if it is an equilibrium reaction, right? By the same hand, concentration of products can also determine if the reaction is going to go backwards or not. Now, okay, so that, okay. Other things that are going to influence the reaction, right? So the speed of the reaction. One thing that is very obvious that is going to influence the speed of the reaction, besides the concentration of products or reactants, right, is the temperature. Because if you have optimal temperature, then you are agitating those reactants to a point that most likely they're going to hit each other and allow the reaction to take place. So more movement, more temperature, then most likely the reaction will take place. Now, the size of your particles is also important. Because let's say you have a good temperature, you have a good concentration. Now, if your reactants are huge, they can try to move around, but they might not have enough, probability of them hitting in the right place to cause a reaction. Smaller ones, smaller particles will give you a better probability of the reaction to happen. And of course, you are looking into catalysts. In the body, those are proteins. So proteins are going to work as catalysts. Okay. What is the catalyst going to do? I was going to see just for a quick review. The catalyst is going to lower the activation energy for some reaction to happen. So I'll just put a quick graph over here, right? So let's say here you have products. Here you have, here you have reactants. Let's say here you have products, okay? Now, let's say that in order for this reaction to happen, you have to reach activation energy, which should be here, right? So a catalyst or protein is going to allow the positioning of the reactants to be just in the right conformation to allow the reaction to happen. Now, notice that in this graph, we have the total energy of the products lower than the total energy of the reactants in your y-axis. So that means for a reaction to happen, it has to be spontaneous, right? Remember delta G, Gibbs free energy? So this thing here Between reactants and products, it should be a negative delta G for the reaction to be spontaneous. Okay? All right. Now, the catalyst is going to lower your activation energy. So the purple dot here is the work of the catalyst. Okay? Catalysts cannot make reactions that are not favorable to happen. Okay? So if you have here some reaction that the products are going to be up here, products here, there is no catalyst that is going to allow this thing to jump from that to that, because that's going to be a positive delta G. That's not spontaneous. Now, when you're going to look at reactions in the body, you're going to see there are many reactions that overall they don't seem spontaneous, but when you break down into pieces, you will see that each individual reaction is spontaneous with a negative delta G, because all of this has to really follow chemical and physical laws. Okay. What would be the effect of a chemical on a chemical reaction if the concentration of reactants is increased? Your answer here is B. The speed of the reaction will increase. Okay. What is making up your body? You're going to have inorganic compounds. You're going to have organic compounds. Now, the inorganic compounds are quite simple to remember. That's going to be water and salts and some acids and bases, right? But those that do not contain carbon, right? So pay attention to this guy here. They do not have carbon, because if those compounds have carbon, then they are considered as organic compounds, okay? So what are the organic compounds? Carbohydrates, fats, protein, nucleic acid, everything else basically that you think in the body, that's all the organic compound. Carbon chemistry is part of your organic compound. Of course, you need both of those to maintain life. Okay. Now, when you look at salts, okay, those salts are inorganic, of course, if they don't have carbon. And when they are away from water, when they are solid and not in water, they make those bonds that are a bit difficult to break. They make those ionic bonds, right? Now, when you drop them in the water, you will see that they're going to dissociate into anions. Okay, so cations and anions. Now, the cations are positively charged, anions are negatively charged. Okay. Now, also make sure that you know of this nomenclature, right? So we're talking about electrolytes and positively charged, negatively charged. Okay. Okay. So this is cool. Acids and bases. All right. So those are electrolytes. They are going to dissociate in water. So fully dissociate, right, in water. Now, definition of acids. Acids are proton donors. We just call it proton. We know it's a hydronium ion. That's the proper name of it, right? We just call it proton. So here, at the end is hydronium ion. So here, when you have something that's going to donate a proton to water to make your hydronium ion, so that is your acid, okay? Here. So here, HCl is a strong acid because it likes to donate that hydrogen very readily, right? So here is your hydrogen. Now, when you dissolve that in water, right, because that's the place where this thing is going to be able to donate that hydrogen, right? When you dissolve this in water, this is when you're going to have your hydronium ion at the end. Okay. What are the bases? Well, if the acids are the proton donors, the bases are your proton acceptors, okay? So here you have something, the salt that is going to dissociate, and this guy here is capable of accepting a proton. So this is what we call the hydroxyl ion. So that is going to come out of a strong base. Okay. So important bases for maintenance of physiological conditions. are bicarbonate ion, okay, and also ammonia. Okay, so bicarbonate ion is very, very important for maintenance of your blood pH, right? And so is ammonium, okay, for waste products. All right. Now, why is it important to have this basic idea about bases and acids? Well, because your blood has to be always kept at a certain pH level. So pH is going to measure the amount of hydrogen that is present in the solution, okay? Now, if you shift the pH from optimal value, you can influence the chemical reactions that are happening in your body, okay? And you can also influence the behavior of cells that are being bathed by this liquid, okay? So what is the pH going to influence? Well, it's going to influence how many things can go through the membrane, so permeability of the membrane is going to also influence how the proteins are folded. Because if you have seen how proteins get expressed and folded, they do that naturally, right? They just do it because they start getting all those amino acids together and those peptide bonds, and they naturally start to fold on themselves. You know, you don't really need a catalyst for the folding of the protein, okay? Now, if the pH of that environment where the protein is being made, if the pH is not optimal, then the stability and the folding of the protein, those can be influenced, usually in a bad way. Okay. Now, the rate of transport through the membrane, that's very much involved with the permeability of the membrane. Other thing that is very important to look at the pH in general is when you start looking at things that have to be absorbed by the body, like, for example, nutrients and drugs and some types of other things that can get inside your body, okay? So things that are important to happen, all the reactions that are important to happen in the body, they require a certain pH for them to take place. Okay. So here, a little bit more on acids and bases. Acidic solutions have a lot of hydrogen, right? A lot of hydrogen floating around, but the pH is very, very low. So the acidic pH goes from 0 to just before 6. Now, neutral solutions, not acidic, not basic, those are exactly at pH 7. So if you talk about pure water, that's pH 7. Okay. Now, when you look at basic solutions, those are also called alkaline solutions. Those are going to have not a lot of hydrogen ions floating around, and they're going to have high pH. Why? Because the pH is based on the logarithm scale of concentration of hydrogen. Yeah, there was like half a semester of chemistry in 2 phrases. All right. I thought you were going to fly through this, right? No. I warned you. Okay. So, now, if you have no idea what I'm talking about, please seek help, because you will need it. Okay. Now, just looking at a big picture on different pages of things that we are usually exposed to. So when you look at the neutral scale, we're going to see the blood is right there. It's like actually very close to milk and eggs, right? Okay, so blood is a pH of 7.4. Then if you look at black coffee, is pH of 5, is a bit more acidic. If you look at red wine, pH of 2.5, is a little bit more acidic, right? Look at lemon, and then this is not water, okay? This is hydrochloric acid. It's kind of misleading that it's looking like in the cup that looks like water. No, hydrochloric acid. Very, very low, very, very low pH. Okay. When you look at other stuff that goes more towards the basic side, you're going to see here hydroxide, sodium hydroxide, pH of 14. It's a very, very strong base. Remember, the base are the ones that accept protons. Okay. All right, so after talking about acids and bases, we need to talk a little bit about buffers. So the buffers, they are going to resist change in the pH of this solution, right? Just because if you want to keep something alive, you better keep it at a stable pH. Right, so when things are happening, acids coming here and bases coming there, you always have those buffers to keep things very much stable. Now, a very important buffer in the blood is bicarbonate system. Okay, so you have carbonic acid here, carbonic acid and bicarbonate, and here you're supposed to have this equilibrium reaction, okay? Because if you look at this, you'll see that your carbonic acid is able to donate a hydrogen, right? And then your bicarbonate here is you can accept a hydrogen, okay? So what's going to happen here is that this is not a strong acid. This is a very weak acid. And this is not a strong base. This is a very weak base. Okay, so when you put things in solution, they can donate a little bit of hydrogen, so this thing is going to pick it up and transform into that form and maintain the overall concentration of hydrogen the same in the solution. Now, what am I talking about? This is what am I talking about. This is what I'm talking. Okay, so let's see what is happening here. If you have a solution, this of course in water, and you are dropping a very strong base in this solution. Okay, so this very strong base is going to collect a hydrogen ion, and this part of the very strong base is going to become water. So this thing disappears. If this thing disappeared, what's going to happen to the total pH of your solution? pH is going to go up, right? The less hydrogen you have there, your pH is going to go up. Now, what's going to happen? Here in the solution, you have this weak acid, okay? Now, this weak acid, what it's going to do is going to donate a hydrogen to replace the one that was taken in your solution. So if you replace this thing here, what's going to happen to your pH overall? To be like nothing, right? Nothing is going to happen because you take this out, you put this in, it's like nothing happened. Okay, so this is the work here in this case of a weak acid that is working as a buffer. Okay, the only time when you're going to see the pH increase in this case is when you run out of weak acid to donate hydrogens. Then you're going to see the pH going up. Okay. What is going to happen to your blood in case you're breathing too much? Yeah.
Your pH is going to go up.
It's a bit complicated question.
I mean, if it's in a response to what's happening, like if you're breathing too fast because of what's happening, then your pH is lower.
Okay, actually, you need to learn a lot more things before I can ask about the blood. Okay, so let's say something. Let's say that your bicarbonate system, okay, is a pH buffer, right? So let's say when you breathe too much, you're going to dump out carbon dioxide. So let's say that's almost equivalent as your bicarbonate system, okay? Overall, your pH of your blood should go a little bit more to the acidic side. Okay, so let's look at this picture here. If your pH in the blood goes a bit to the acidic side, that is called acidemia, acid in the blood. Emia usually is stuff in the blood. So if the pH, if the concentration of hydrogen ions goes up in the blood, your pH goes down, so you get acidemia. And look at the range, it's like 7.35. It wasn't very far off from the normal blood range of pH, okay? Now, let's see if the opposite happens. and you have a lot of base. So that means very little hydrogen in the system. So now you're going to have a lot more of a alkylosis, or you have a lot of alkaline environment. That means your pH should be up. That means this is a problem for the blood as well. Okay. So really, your buffer system in the blood that has to do with bicarbonate, right? Your buffer system in the blood is what is going to keep your blood at this normal pH so that you stay alive. Okay, I know this is like really flying through, but just for you to have an idea of how important that system is in order to keep all this liquid in a range that is optimal for enzymatic function. Okay. Oh, here you have another question. Right. A buffer will release hydrogens if the blood pH, what happens? Something happens to the blood pH, and then you have to release hydrogens. A. Everybody got that? Okay, so if you have basic, means a lot of OH minus, then your buffer will release hydrogens. All right.